Hello, I'm Carol, and today, for the last time of this course, I will be exploring a very important transmembrane protein, the gap junction beta 2, also known as Connexin 26, CX26, CXB2 human, and so on. I would start my presentation explaining how important it is to cells to connect and communicate between themselves. So we know that this intercellular signaling is one of the most essential properties of multicellular organisms like me and you. And this is how multiple cells are going to act as a part of a tissue, an organ, a system. And the gap junctions are specialized regions of the cell membranes that contain some channels that allow the passage of some molecules, ions, and this is super important for a lot of biological processes. So the Connexin 26, this is how I'm going to address to it. The main role of this protein is to work as a channel of a potassium and other small molecules. And of course, there's a lot of connexin proteins, a lot of them, and sometimes they work together, so a mutation in one can impact the other one as well. We'll explore a little bit more about the molecular structure in general. The Connexin 26 have a positively charged cytoplasmic entrance, a funo, a negatively charged transmembrane pathway as well. And remember that potassium is positively charged. It has an extracellular cavity. So the pore itself works like a funnel, which is formed by six amino terminal helices line the wall of the channel. This narrow funnel determines the molecular size restriction at the channel entrance, which is really small molecules. Okay, we got it. This protein is super important, but what genetics is behind it, right? The gene that codes for this protein is called GJB2, and it is found in the 13th chromosome in the Q arm. And after this gene being transcribed, translated, and the protein folded, now the protein needs to go to a compartment to where the protein is going to perform its role. And as I said, it is a gap junction. So where this protein is located? In the cell membrane, right between the two cells. Even though we can find this protein throughout all the body, it plays a huge role in the inner ear. Okay, so if it is present in there, does it have something to do with hearing? Spoiler, yes. So basically, hearing requires the conversion of sound waves into electro nerve impulses. This process is super complex. I'm going to spare you of all the biophysics behind it. So remember when I explained that the connexin transports potassium? Exactly. So hearing includes a proper maintenance of the potassium levels in the inner ear. If there's any problem with this gene that codes to this protein, the hearing capacity can be affected. The connexin 26 is present in a lot of tissues and also in the skin, which the malfunctioning of this protein can also lead to skin conditions. But I want to focus in the hearing loss conditions, which is especially interesting for me because the same gene can lead to two different pattern inheritance. So if we look into this first mutation, the DFNB1A, the inheritance will be autosomal recessive. And if it is the other one, it is dominant. This proves us that mutations can have so many different effects, even though that it's in the same region, in the same gene, but they are what? Different variants, different mutational events. So it's a total new effect. That's fascinating to me. So it is important to know that the connexin 26 alterations are responsible for at least 20% of all genetically hearing loss. That's a lot. That's a lot. So on the cochleas, now the part of the inner ear, in the junction gaps that exist in there, when the proteins is missing, something is happening, the junction gaps just fail to work and the cochlear hair cells die off, which leaves the body incapable to translate sounds into nerve impulses, and this person have a significant loss of their hearing capacity. In terms of testing for this condition, specifically hearing loss, there is the possibility to make a polymerase chain reaction, which will analyze the regions of this gene and check if there is some common mutation of some case of genetic deafness and look for a specific mutation. Also, they can make conduction tests that will stimulate some sounds in different ways to see if there's some response. Thank you so much to have watched so far. I learned a lot and I naturally love talking about this stuff. So if everyone is curious to discuss more about molecular biology evolution, please send me an email and I will be happy to get in touch with you. So bye and see you in another opportunity.